Okay, so good morning, everyone. Owen, I don't know if you want to say a few words to introduce everyone while I'm finishing setting this up.
Okay, well, thanks, Jeff, for setting everything up, and thanks everyone for joining us again for the March project meeting here. And one thing that we've had another successful joining of the program. I'd like to recognize on the agenda here, hopefully between 10:15 and 10:45, hopefully they'll be able to join. But Alexi and Greg were people we were introduced to, and they're going to be hopefully talking about briefly some of the inhibitors for the RIPK2. And we continue to bring more and more parties into this collaboration, so we're really happy to have them join, etc. Also crossing the pond today with us, Alex, welcome to the other side. It's actually warmer here in Toronto than I believe in the UK.
Thank you, Elliot. Yeah.
Welcome everyone. And with that, I won't do too much of the introductions. I think everyone has been on the lines before. So why don't we try and get going? We're going to try and really focus on ending this at noon because everyone's we know is very busy. And we'll move forward from there. So thanks, Jeff. Why don't we start running through?
Okay. It looks like we're just having a little bit of, Alexi's having a little bit of trouble getting in. Let me just send him a quick email. Okay. Just give it 2 minutes to get Alexi in. Greg, are you logged in? Greg?
Hello? Yes, I'm on.
Yeah, okay, great. You're logged in. So, yeah, Alexi just seems to be having some issues, so hopefully he can resolve that. So, just to get the meeting started, this is the agenda here up on the screen. I hope everyone can see the slides. Please let me know if you don't, and we can try to address that. We're still getting used to this format of WebEx that allows us to record and sync the sound with the visuals. So it's a new system for us. We tried it out last month. There were a few kinks, and I think we've worked out most of them. This seems to be smoother already, but we're still dealing with some issues. So let me just start the slideshow here. Okay, so this is the agenda. I'm going to go through some of the action items from the previous meetings, just very quickly. And then, as Owen mentioned, we have Greg and Alexi on board who have worked on the very same scaffold from which our lead comes from, but they've gone after this scaffold for its off-target activity for us, the RIPK2 activity. But in the course of their work, they've closely monitored the ALK2 potency, and so they've come up with what I think are some interesting SAR and some interesting observations. So they've kindly agreed to share that information and to give a presentation today. Alex is going to talk about the development of some cell-based assays, and we just got some data on the initial 30 compounds. It looks quite exciting, and I'll talk a little bit about that in this session. This section might actually be a little bit longer because I've got a little bit more data than I anticipated having, but that's okay because I think Owen at the end will be slightly shorter than what we've indicated here. David McLeod from our group here at OICR will talk about the chemistry that we've done here. And Sue will give an update of the Charles River chemistry and some of the ideas that they have for moving forward, and then we'll have we'll get to hear Owen talk about some administrative items, and we'll summarize at the end. So just to bring everyone back up to speed and to let everyone know what's going on with this project in terms of the Cancer Therapeutics Innovation Pipeline grant that was one of the main sources of funding for this project. So we have funding for 24 months. We are now about 3 months in, so the first quarter is done. And in months 1 to 18, we're supposed to essentially be doing SAR in a couple of different series. The lead series, around 75 new molecules. You'll see today that we've already submitted 35, so we're well on the way to meeting that deliverable. But I think it's going to take more than that to get to the compound that we want. A backup series that is, there's multiple choices at this point, but I think having the Institute for Cancer Research, ICR, involved, they've got an interesting series of molecules that looks like it's a great potential for a backup. So, I think only talking about, sorry, the backup's actually down here, but this is some X-ray studies. We're looking at the MDCK MDR1. This is getting brought in-house, and the tech transfer has already occurred. So we're working on getting that in-house. We're measuring selectivity. We're looking at some cellular efficacy and mechanism. So basically, we're only about 3 months into this right now. So I think we're in good shape, and I think you'll see by the end of this meeting that we're starting to get some really valuable data that will help guide the SAR and to drive the project. So this is the ALK2 project flow scheme. I've updated this a little bit based on some of the data that we have just recently received. So we still have predominantly Charles River and OICR following the pyridine series of molecules. The quinazolinone series at ICR is still temporarily on hold. We're working on getting them on board to support the project. But as of now, they're still kind of sitting on the sidelines. But we had a good discussion yesterday with Raj, and I think that's going to happen in the not too distant future. As the compounds get made, they go into an ALK 1, 2. They will go into an ALK 1, 2, and 5 enzymatic assay at RBC. So our reaction biology has graciously agreed to test our compounds. We just recently got some activity against the ALK2 DIPG mutants, and it seems like the SAR tracks very well with wild type, so, we don't have to track these on a regular basis, but certainly, moving forward, we will want to spot check key compounds against these mutants to make sure that we're maintaining activity against the broad spectrum of mutants that we see in DIPG. Alex's group is looking at some cellular assays. He's got some nanobread data to show today that I think is very exciting, so I'll leave that. We've also had some luciferase data previously, but it looks like the nanobread is going to be the way to go. Alex's group is also generating X-rays, X-ray co-crystals for some of the key compounds, and we'll see some of that data hopefully in the not too distant future. Here at OICR, we're characterizing the compounds for the liver microsome stability, and as I mentioned, we're bringing in the MDCK, MDR1 model because this is a project for which we need brain penetration, so this is a really good model to look at that. And then, we also want to look at phosphosmad and proliferation in really the DIPG patient cells. I'll go through some data later, and this is really, I think, key for showing or predicting whether or not a compound is going to show efficacy in an in vivo model. And so getting this data on key compounds is going to be something very, very important moving forward. So RBC not only agreed to test, compounds against 3 kinases per month, but they've also agreed to look at a subset of compounds against 5 other kinases each month. And so the ones we're kind of looking at now are potentially these ones, PDGRF, potentially alpha actually, and one of the ALK2 mutants perhaps, and some of these others for which the lead compound showed some activity. Molecules will then move in vivo, and we'll do some of that work here at OICR, at least the initial plasma exposure, potentially some brain exposure, but that still needs to be worked out. Really depends on how many compounds we move forward. And then moving beyond that, I think we still need to figure out exactly where we're going to go and where we're going to have all of that, the more extensive work done in terms of efficacy studies and some of the advanced in vitro ADME that will be necessary to really select a candidate from this project. One new and noteworthy thing that I did notice is that Blueprint has actually selected a candidate from their ALK2 project, Blue 782, to treat FOP. They presented some data that makes this compound look pretty promising. We have their patent, and Sue will talk later about how the synthesis of some of the representative analogs from that patent is going. I think we'll be able to generate some data on some of the compounds from that patent very soon. And with that, I'll move into the action items. So as I mentioned, Greg and Alexei have worked on RIPK2, but starting with the same series of compounds that from which our lead comes. They've shared that data with us. We've executed some MTAs and generated a list of their compounds that we think might be interesting for studying in ALK2. We also agreed that they could test some of our compounds against RIPK2 if they're interested, so they will be following the SAR carefully as well. And today, as I mentioned, they're going to tell us about their RIPK2 story. So I won't move beyond that, but we're certainly glad to have them on board, and I think their expertise with this family of kinases is going to be valuable for driving the project forward. There does seem to be a disconnect between the targeted engagement and DIPG cell line proliferation data. This I'm going to talk about a little bit later, and I think maybe we'll see that there's not such a disconnect as much as we might have thought at one point. And so, I think I'll go through some data later that will speak to this a little bit. And then this initial set of 30 compounds that is going through the cascade. So we've had the compounds tested at RBC. We just got the cell-based data from Alex, and I will show some SAR later in the presentation once we get into that part of the meeting. In terms of action items from the February 6th meeting, Sue and I talked about the need to coordinate between Charles River and OICR to ensure that there's no duplication of effort, that we're not making the same analogs. So Charles River has enabled us to access their web portal, which allows us to share documents back and forth. So we've deposited files that contain our ongoing work from our internal chemistry effort, and Sue has also put work documents in there that share the Charles River data. And so, these lists will be updated and shared on a regular basis, and I think that should really help eliminate any possibility of duplicating effort. And, so far there hasn't been any issues. Shipping compounds to RBC for testing needs to be coordinated across sites. So this is as well as something that Sue and I have discussed and looked at possible scenarios. It doesn't seem to be an issue for the time being. I think Charles River is still working very hard on the chemistry that they've set out to do to generate analogs from these patents. So they don't have a tremendous amount of compounds to ship at this point, but they do have a couple. And so we're working out what the best arrangement is, if they send it to us and then we ship it to RBC, or whether or not they just wait until they have a number that makes it worthwhile to send to RBC. So this is still being worked out, and Sue and I are discussing this. And then, just to remind everyone that each compound should be assigned M4K numbers, and we're still looking at getting the database up and running. Our database here is up, and M4K numbers are being generated automatically. There's still some work to do in terms of data retrieval and data display in a convenient format, and Genna is working on that. And that should be up and running in the not-too-distant future as well. And that's this compound progression.
Is that database going to be open to Charles River and Oxford site?
We will upload files that you will be able to incorporate into your own database. I suspect that that's going to be the easiest way to do that. So we can send SD files. We will be able to send Excel files as well and kind of display the data. And
Perfect.
yep. Yeah.
As far as we're concerned at Charles River, we have a database set up, and when we get the SD files from Jeff, then we will just upload into our own database.
Right. Okay. I'm not sure if Alexi's been able to log in yet. Let me just double check on this. If not, then I'll get Angela involved. Looks like. Alexi, are you on the line?
Maybe we can ask Greg to start presenting.
Yeah.
Yeah, I can start. Are you going to load the slides? Okay, there we go.
Yeah. I'll move them forward as well. So just let me know when you're
okay.
when you want me to advance them.
Yeah. So when I put this together, I really put it together to focus on the ALK2 part. I mean, I'm happy to answer any questions about the RIP2. I'll show you the RIP2 data, but the focus is going to be on ALK2 since I thought that would be most useful. So this is just an outline of what I'll talk about today. So I'll just give you a little bit of background on how I got connected in ALK2 and also how I got interested in RIPK2 and then sort of how it came together. So the interest in ALK2 started back when I was at Brigham and Women's Hospital. I was at a lab, just a little bit of background, that was focused in drug discovery, but primarily in neurodegenerative diseases. 2006, they decided, partners decide, which is a organization sort of that over several of the Harvard hospitals, including Brigham, Mass General, and several others, was interested in expanding what we were doing, which was sort of a high-throughput screening center, med-chem, drug discovery center, to expand that beyond neurodegenerative diseases. And so they supported an effort for about 3 years. And one of the projects that came in through Paul Yu, who was a fellow in Ken Block's group at MGH, and they were working with Randy Peterson, and they were interested in this bone morphogenetic protein pathway. And when they came in this to see me with their lead compound, they were initially interested in vascular calcification, but this sort of, because Ken and Paul are both the cardiologists, but they sort of switched very quickly to FOP. And also through this project is when I first started to collaborate with Alex Bullock in getting crystal structures on some of the compounds we were developing for ALK2 and for FOP. And so the story is, Paul and Ken and Randy were screening in a zebrafish model, and they had, and a guy named Charles Hong, who was also there. They were screening in zebrafish looking for things that sort of had an effect on the BMP pathway, and they pulled out this compound, which was referred to in the literature as compound C, and they renamed dorsomorphin. And this is when they came to see me about, what I might be able to do with them in terms of optimizing this compound for, at the time was for inhibiting BMP signaling, but then we fairly soon realized that the target was ALK2. And so we worked on this for a while and developed sort of the first compound in this series that was potent and was selective over the TGF-beta family, and that was this compound LDN-193-189. And later we've made other modifications, and there's still a lot of interest in this compound series from the perspective of FOP, although in my own lab I'm really not working on this series at all for FOP. So that was how I got involved in ALK2 and have been involved in various aspects for a while with Paul and Randy. And then on the next slide I'll just introduce how we got interested in RIP2. So this I should mention also, yes. So then we also, after the 189 series, Alex's group had found this compound K0288. This is what I refer to it as the pyridine series. And we got interested in also looking at the SAR of this compound, and we started developing it. And one of the compounds that emerged was this compound 214117, which retained the trimethoxy group. It really just switched the solvent-exposed region. And in some ways, it was better if we got rid of the hinge-binding amino group, although I'll talk a little bit more about that later. You do lose potency, but we seem to gain a little bit of selectivity. So that was sort of in the paper we wrote, we sort of highlighted this compound. Although with the amino group, it's still, probably a little bit more potent, maybe a little less selective. So that was the work in ALK2. And then on the next slide, how these two end up coming together was an interest. If you can go to the next slide, is in this protein family, and this actually predates interest in ALK2, which is an interest in these targets called receptor interacting protein kinases. And when I first went to the Brigham in 2001, the very first project I started a collaboration with was with Junying Yuan's group in the Department of Cell Biology. And Alexi was a postdoc in her group at the time. And they were looking at another pathway. They were looking at what they believed would be sort of a programmed necrotic cell death pathway, and they had screened and were looking for compounds that inhibited this process, and they had pulled out a couple different series of compounds that we worked on with them, one of which is shown here. It's sort of the lead series, sometimes referred to as the NEC1 series. And several years after we started, the target was identified as being RIPK1. And we've optimized a number of series for these compounds, and I just show here a kinase profile. We were able to develop actually quite selective inhibitors of this target. We worked on this for quite a while, trying to understand some areas of disease that might benefit from inhibiting this particular kinase and that were dependent on this regulated necrotic cell death pathway they term necroptosis. And but over the last couple years, Alexi and I have become more interested and sort of shifted our interest to a related kinase called RIPK2, which is not in the cell death pathway, but has more of a role in inflammation. And so we began to work on, in the last maybe 2 or 3 years in RIPK2. So if you go to the next slide, I'll sort of show you how these sort of came together. When we were initially looking for a lead in the area of RIPK2, we thought about maybe looking at these type 2 DFG out inhibitors as maybe a good lead structure. And we put together this small little panel. I mean, I showed 2 compounds, but the panel was a little bit bigger than this, of known type 2 inhibitors, and wanted to see if we could find something that might inhibit RIPK2 from this set. And we pulled out, I show you here, 2 compounds, the well-known compounds ponatinib and regorafenib, which were able to inhibit RIPK2. We also were interested in this set of compounds for examining ALK2, thinking about the fact that the first 2 series we'd worked on were type 1 inhibitors. Would it be possible that one of these type 2 inhibitors might inhibit ALK2, and it would be a different mode of inhibition, and maybe it would offer some increased selectivity or whatnot. And we were quite surprised to find that none of these compounds inhibited ALK2, which was sort of disappointing for us, but also started to indicate that, ALK2 is sort of interesting in that it doesn't seem to be able to potentially adopt a DFG out conformation. And so this was one potential way to enter into RIPK2. But another way we thought about, as shown on the next slide, has to do with this idea of kinases being able to go from, DFG in to DFG out conformations. RIPK2 obviously can, and this is a paper from the group at GSK where they, again, solved some different crystal structures with some different inhibitors and saw varying degrees of movement, where they, for example, the alpha C helix, suggesting, that you could potentially inhibit RIPK2 as, type 1, potentially a type 1.5, where you get some movement of the alpha C helix. And as we had shown with ponatinib and regorafenib, you could also inhibit it via type 2 mechanism. And we started thinking on ALK2 that may not be possible. Maybe you can't go all the way over to a type 2, but could you potentially go to this type of 1.5 inhibitor where it starts moving things, but it doesn't push it all the way to a DFG out inhibitor? And might we use this to explore RIPK2? This was where the focus came in RIPK2. So the question was what kind of scaffold to start with to try to examine this idea. And we had known, and if you go to the next slide, we had known from both the LDN series and this the pyridine series that the ALK2 compounds always seem to bring along with it very potent RIPK2 activity. And so we thought about the two series we had worked on. We thought, well, which one of these might be interesting to pursue as something that we might try to convert from a ALK2 inhibitor maybe to a RIP2 selective inhibitor, and could we use it as a scaffold where we could look at trying to explore these different conformations of RIPK2 to see which one might be beneficial in terms of inhibiting the kinase, and also, and turns out more importantly probably, the signaling pathway involved with RIPK2. And so this is how we got back to the pyridine theory in its relationship to RIPK2. We thought this might be a nice scaffold to pursue, and could we do it in such a way that would potentially generate some interesting RIPK2 inhibitors. And I think from the point of view of today's discussion, I think we found some SAR in there that may help you guys think about, and maybe not repeat some of the things we did in relationship to ALK2. So we used ALK2 as our counter screen, thinking that these two kinases, in many ways, and with at least two different series of compounds, seem to be inhibited by both. And if we could generate selectivity versus ALK2, maybe that would translate into broader selectivity versus, more kinases. And so that was sort of our rationale for picking ALK2 as our counter screen for our RIP2 program. And so this is just a crystal structure that was of this pyridine series. And so one of the things we wanted to do was see if we could try to figure out a way to manipulate this compound to start engaging areas over here around the alpha C helix or the DFG portion of the molecule. So on the next slide, so the idea was, could we take this scaffold and figure out a position on the molecule to start introducing groups which might, in the best world, would start moving these pieces like the alpha C helix and interacting with the DFG, and would this potentially give us some interesting leads into RIPK2 inhibition, and also might translate to good, cellular potency and eventually in vivo activity in areas where RIPK2 has been implicated. And for us, it's turned out to be primarily right now in CNS. There's a lot of people interested in this target for IBD, inflammatory peripheral inflammation, but we've been thinking about it more from the neuroinflammation area. And I should mention, in the FOP area, it's, so people ask me, why do you want to make a selective compound, and separate out RIPK2 and FOP? And I think in FOP, there's not a real good reason to do it. I think a lot of people looking at FOP don't really care that much about the RIPK2 inhibition coming along. And again, I don't know what the opinion is for DIPG. Maybe it looks like you're putting that up as a counter screen for RIPK2. But again, I don't know whether that's a problem or not for DIPG. And I think a lot of people feel in FOP it's probably not. In fact, one of the reasons I'd like to separate out and make a selective ALK2 compound is I'm curious about, what the potential role of inhibition of RIPK2 is in FOP. It's possible that actually it's best to have both an inflammatory inhibition in addition to, inhibition of ALK2, but that's sort of debatable. And one of the reasons I'd like a selective ALK2 compound is I think it would be interesting in FOP to look and to see if efficacy is, enhanced by having RIPK2 inhibition, or is an off target and you would prefer not to inhibit RIPK2. But our design was to look for a compound that might be known to inhibit through this type 1.5 mechanism, and look at the fragment that might be in that molecule and try to combine it with our scaffold on the pyridine scaffold. And that was sort of where this hybridization approach came from. So what we needed to do was identify a compound that would be this type 1.5 inhibitor and find out, basically what piece of that molecule might we graft onto the pyridine structure and examine it. So we went looking in this links database at Harvard for compounds that might be interesting to consider as one of the fragments. And so on the next slide, I'll show you the compound that we chose, and that was, again, looking through this database, we found this compound, this PLX compound that, its main target is in BRAF, but it also shows this very potent inhibition of RIPK2 at about over 90% inhibition at 10 micromolar. And shown on the right is the crystal structure of how it interacts with the DFG and pushes the alpha C helix out based on this sulfonamide group that's present in the PLX compound. And so the next stage for us was to start overlapping these structures, and they're shown on the next slide. So when we overlap this BRAF structure on the top portion, the PLX compound shown in the cyan, that sulfonamide sort of overlays with, and we're overlaying this with our 117 structure with the ALK2. This sulfonamide sort of lines up with the methoxy group in the 4 position of the phenyl ring. So that trimethoxy, it's the one in the center that it lined up well with. The structure on the bottom we then docked this structure into a RIPK2 structure, and then aligned those structures again with the PLX BRAF structure. And in this case, it tended to align better with the methoxy group in the 3 position, not the 4 position. So we thought, okay, maybe the first thing we need to do is we need to generate 2 derivatives of this structure where we install this sulfonamide in the 3 position and the 4 position and see which of these 2 might inhibit RIPK2. And again, use ALK2 as our counter screen. And so this became the strategy on this slide, was to sort of take these 2 and use the fragment, the sulfonamide fragment, and bring it into the LDN 117. Now, when we were doing this, my initial assumption was that based on the prior work we did on the LDN 117 scaffold, is that we might completely lose ALK2 activity, because we knew before that these trimethoxy groups seem to be quite important for activity. So when we initially designed this, I thought, okay, we may completely lose ALK2 activity. I didn't really expect it to be retained, because we thought that, okay, those methoxy groups were really not replaceable, particularly by switching it from a donor group that's fairly somewhat hydrophobic to this hydrophilic sulfonamide that we might really just not retain ALK2 inhibition. So we made the compounds. I'll show you the SAR here. With 117, this is the parent compound. And then the first 2 compounds we made were just placing the sulfonamide either in a para or meta position. We retained 1 methoxy group. So all these compounds in this table have this methoxy group. And we had known from the SAR initially that for ALK2, we could tolerate not having an R1 substituent. So we thought, okay, we could go from 3 substituents to 2 and put that sulfonamide in one of 2 positions. And when we did that, when we found that if we put it in the para position, it lost all activity, both against RIPK2 and ALK2. So it didn't like that. But if we put it in the meta position, that it actually looked pretty good against ALK2 inhibition, 31 nanomolar, and it gave us modest inhibition of RIPK2. So it was a little surprising that it would tolerate the sulfonamide just based on our prior work, but nonetheless it did. And it also showed us that it needed to be in this meta position. And the next set of compounds is basically just varying. The next thing we did was really just vary the size of the R group on the sulfonamide, and didn't find tremendous SAR. Methyl, ethyl, a little bit longer. Propyl, we made it branched. We also made it aromatic with phenyl. And what happened was we retained all the ALK2 inhibition, but it made the RIPK2 a little bit better by going to larger groups. We also switched, you'll see here in the column X, we also quickly switched back to the better hinge binding group, back to NH2 as opposed to methyl. And that also was particularly helpful in RIPK2. And so, again, changing the sulfonamide didn't really do much. It retained ALK2. RIPK2 got a little bit better. There is one interesting compound I'll point out here at the bottom, which we've not really focused on, but it's sort of interesting from the point of view of getting selectivity to go in the direction for ALK2. And that's this compound 24. When we changed it from a benzyl sulfonamide, from a phenyl sulfonamide to benzyl sulfonamide, we seem to get a preferential inhibition of ALK2. This probably needs to be examined in more detail to really conclusively say that this is a direction where one could go to potentially get rid of ALK2 inhibition. But it is sort of interesting. And at the time, again, we were focused on RIPK2, so we haven't necessarily pursued that to a great extent. So on the next slide, I just show a set of compounds which begins to now examine the other two positions, the R1, what we call R1 and R2, with this propylsulfonamide. And I think from the perspective of ALK2, it shows that these groups in these two positions are quite sensitive to modification. So if we, for example, in 43, if we reinstall a methoxy group in the para position as R2, we lose some activity against ALK2, although it still was, 60 nanomolar. Again, it doesn't have much effect on RIPK2. But when we change it to methyl, then we actually have a lot of problems both with RIPK2 and with ALK2. And if we remove, for the next three compounds, if we start removing the group at the R1 position, this methoxy group, then ALK2 really suffers. And the theme is going to be that methoxy group, it really tends to like that methoxy group in ALK2. So you can see that these last three compounds, whether we have methyl or methoxy at R2, if we don't have methoxy at R1, the RIPK2 activity is just not very good at all. Okay, on the next slide, a few other changes we made. Again, this is compound 43 in the box, where you have a methoxy at this R1 position. Again, for RIPK2, we found that we could change that. The next two compounds, we could change that methoxy to other things like fluoro and chloro and still retain potent RIPK2 activity. But again, ALK2 really suffered. It just doesn't like not having that methoxy in that position. Compound 52 is sort of interesting, because if you compare it to 43, we thought, well, what happens if we take these two ethers, this R1 and R2, and we tie them together into this methylene dioxy? And as you can see, it really does not like it at all in ALK2. No inhibition. And the last two compounds are sort of interesting. They're not sulfonamides, but again, they show that as long as you have this methoxy in R1, and you may or may not have a methoxy in R2, you can put other things in this R3 where the sulfonamide is for RIPK2, such as just a fluorine atom. Again, some of these may be useful from the point of view that a lot of people are worried about this trimethoxy for metabolic stability issues. I think you could at least get rid of one of them with a fluorine, and I wouldn't be surprised if you could keep that fluorine, get rid of the R2 position. With a hydrogen, you might still retain good ALK2 inhibition, and now I've gotten rid of two of the three trimethoxy groups, two of the three methoxy groups on that ring. The last compound here, 23, is also sort of interesting, where we put a benzyloxy group at R3. So instead of sulfonamide, and again retains pretty decent ALK2 inhibition, but again is sort of an interesting data point because it also starts to give us selectivity over the RIPK2. Now again, I think this has to be explored in more detail, but it sort of reminds me, it's sort of similar to what we saw when we put this benzyl sulfonamide in this position. And this ether might be interesting. One concern about the sulfonamide is we do introduce another hydrogen bond donor. So again, even from the perspective of CNS penetration, maybe not the best, although we're looking at that. And from DIPG, maybe going not so much the sulfonamide route, but maybe this type of ether route where you begin to see selectivity at ALK2, you wouldn't necessarily be, other than adding larger molecular weight, you're not really introducing a lot that would hurt you from CNS penetration. And it seems to give some selectivity. So I think it's sort of an interesting data point for ALK2. So that's sort of a summary. I mean, we've made some other compounds. I think Jeff has the entire list, but I just sort of picked through some of the structures we've made to sort of highlight what I think is maybe the most important data points from the perspective of ALK2. And if you look on the next slide, I think is our summary slide. Really from what we've done, and I don't think, I think a lot of this was supported in our original paper, but maybe we've expanded it a little bit, where R1 really does like to be methoxy. I should mention that we have made it even ethoxy, but we also have the R2 as ethoxy. It was just easier to make them both the same alkyl group. That compound is not active. And so what we've never done is gone back and made ethoxy with a hydrogen in the para position or a methoxy in the para position. Again, that it may be tolerated, but right now the thought is that it probably has to be fairly small. We haven't looked at like trifluoromethoxy, which might be another potential compound to make if you're still worried about metabolic stability of a methyl ether. R2, again, for for ALK2, it seems like hydrogen and methoxy is good in this position. And then R3 seemed to be the one that we could vary more so than I thought we could based on our prior paper, and that is we could, we didn't need it, we could remove it, could have a methoxy, we showed that we could put a fluorine in there, and then all these compounds where we put a sulfonamide in many cases, as long as R1 and R2 were the right things, then it didn't seem to care what this R group was on the sulfonamide. And also I didn't put on here the phenoxy. So the phenyl ether also I showed there in that last compound could be put in. So this part seems to be the most amenable to change, which again at least gets rid of one methoxy from the point of view of stability, and looks like you could put things in there that might not hurt you for BBB penetration. We haven't really looked that much at the solvent-exposed region. We've looked a little bit at that region, but again, primarily from the point of view of RIPK2. And going forward, we primarily always kept the central ring as a 2-aminopyridine. So I showed just here, this is again the original crystal structure of the pyridine series, just where these groups are. And I think from this structure, this R1 being methoxy is, this is the way we think it will go into ALK2, where that methoxy group is the one at R1. This bond can rotate. It could be R1 and R3 could switch, but we think that's the R1 group, R2, and the sulfonamide, based on the structures from for the RIPK2, the sulfonamide would be an R3 position, which also might explain why that group is much more tolerant to changes based in its position. We don't have a crystal structure on these structures right now on ALK2. We do in RIPK2, but not ALK2. And so the question is do we see changes in the structure going back to sort of this original point of ALK2 liking to be a type 1 inhibitor, RIPK2 able to go all the way over to a type 2 inhibitor, whether some of these changes like adding the sulfonamide is getting us to some intermediate state in ALK2, we don't know at this point. We do know in RIPK2 that it is perturbing some of these regions in the right, the alpha C helix. So it's possible it's doing that in ALK2, and maybe for ALK2, maybe that's all you can do. Maybe that's the most you can ask. You can't really generate it or force it over to a type 2 mechanism. The data, at least what I've seen, seems to suggest that. So that's basically the presentation that I had. And,
that's fantastic, Greg. Thanks so much for that. I think, like I said earlier, the insights that you can provide, I think, are really, really helpful. I think, I think you'll be interested to see some of the SAR that we've generated today. And unfortunately, it doesn't look like Alexei has been able to get in.
Okay.
But, that's okay because I think we're kind of right on schedule here anyway. So maybe we can have Alexei's presentation at the at the next meeting.
So Jeff, sorry, we have one question from Venice.
Yeah, I don't really know what he was going to present. I think if it's on RIPK2, I mean, again, I don't know how interested the group is in RIPK2 per se relative to DIPG, but that story is very interesting, but very different than I think what is important for ALK2.
Okay, perfect. Yeah, no, I mean, I think we'll talk to Alexei offline and see what makes the most sense. I think there's a question here for you, Greg.
Yeah.
The question is very interesting idea, of course, and since this idea have been inspired by BRAF inhibitors like vemurafenib and abrafinib, do these compounds hit BRAF? Probably would be good to check that.
BRAF, yeah. So we have profiled one of these compounds against a small panel of 97 kinases. BRAF was, I don't think, the top one. I'd have to go back and actually look at the list to see where BRAF was in that panel. So again, we've done
also
this
an
one.
inhibitor with sulfonamide and benzylsulfonamide kind of.
Yeah, right. I mean, that's where the PLX compound started was with the BRAF.
All right. Thank you.
Can you comment on whether you looked in other ALKs, selectivity over other ALKs? Do you have any insight on that?
For these compounds, we have not. In fact, one of the things we're gearing up to do is to provide samples of these compounds of interest. Jeff has given me a list to really have them tested. We have not even looked at them in cell-based assays to any real extent for ALK2. I mean, we've been looking at them in a cell-based assay related to RIPK2. We do know that they do have activity there, so we're pretty confident that they get in the cell and there's no issue there. But we have not looked at them to much degree in any assay related to ALK2. So I think the idea is provide us some samples that Alex can look at in his various assays to see.
Yeah, my question was more about not in-cell activity, but selectivity over other ALKs.
Oh, yeah. So again, we have not looked extensively at that because, again, our focus has been primarily on RIPK2. So that would be something else that, it would be great if we send these compounds over if you guys could look at that. Because, again, from our perspective of RIPK2, we would not want to be inhibiting, ALK5, for example. So again, the hope is that we would retain good selectivity at least over something like ALK5. That would be good for us from the perspective of RIPK2.
Yeah. I'll move on to some of the cell assays we've done.
Thanks, Greg.
Okay, yep, no problem.
So as Jeff alluded to, we've got two activities here in Oxford. One is to support some crystallography, and the other is around cell assays. I don't have a structure to report this month. I think hopefully next month we will. 3 or 4 of the compounds have gone into crystal trials. This is just one of the hits for one of the compounds. The crystals are a bit of a funny shape, but we've seen that before with this kinase. From a core screen, we had 3.5 angstrom diffraction. These are now going into fine screens, and hopefully in the next week or so we'll have tested new data sets to see where we are. So that's a work in progress. And please let us know, if you have those compounds you rank as the most high priority for structures, then point them out, and
we'll definitely
do that.
Later today.
Yeah. Okay. So I'm going to have a number of slides on cell assays, but I thought I'd have one slide just to kind of summarize where our assays kind of lie. So we've been working with Promega on a nanobret assay, which is a sort of real direct target displacement of compounds in the ATP pocket. So that's really at the kinase. Jeff, does this have a pointer as well? Is that the black button?
You should just be able to just roll.
Oh, okay. Yeah. So the other potential assay is we can detect the phosphorylation of the substrate in cells, the SMAD158. And then we also have a downstream transcriptional reporter. So we have luciferase reporters based on the SMAD binding elements. And of course, in this pathway, we have a number of ALKs that signal with BMPs, and a number of other alpha ALK5 that signal with TGF-beta and actin. And they have different promoter and SMAD substrates. Okay. Next slide.
I can.
Oh, okay. So the nanobret might be well known to you, but basically we have a fluorescent tracer that binds to the ATP pocket attached to a kinase inhibitor. The kinase, so out here, we'd have a plasmid that's fused to a nanoluciferase protein construct. When you put in then your drug of interest, you then displace the tracer. And this is some of the data with ALK2, so you get this nice decrease in signal. So this would be sort of triplicate on the same day. Nice assay, and then these IC50s agree quite well with what we'd already seen with other assays like luciferase. So this was this is one of the compounds that Greg had kind of gone through. Okay, Promega on their website they have ALK2 and ALK4. Actually, I'm not sure if the ALK2 is on there yet, but we've been ALK5 they haven't put on their website as where a trace is available, but we have so many compounds that kind of interact between these families. I'm sure we can get that up and running soon. ALK3, they've just said they've just found a good tracer for that one, so they can ship that to us. So the only thing you need to do is clone the luciferase onto your kinase of interest, and then have a tracer that's sort of relevant for your kinase. Okay. This is data from another assay, so we can fix cells in a 96-well plate. In this case, we're not doing any transfection, we're just looking at the endogenous protein. And then we fix cells with formaldehyde, doing immunofluorescent staining for the substrate phosphorylation. So for example, this would be without inhibitor, you get the high signal from the phosphorylated SMAD. When you add the inhibitor, 500 nanomole, as it's down here, you're inhibiting, and then this is just quantifying it from Celigo cell imager. Okay, and there's about 30,000 cells that are imaged in there. So actually you get quite nice curve data from that. So this is the same compound as the previous slide. As you can see here, 53 nanomolar comparing to the nanobret of 25 nanomole. Okay. Well, you'd see this phosphorylation within 30 minutes to an hour of your BMP treatment. The procedure to obviously fix the cells is a bit more laborious than doing a readout with a nanobret or a system. So we wouldn't use this as a assay of choice for routine SAR, but if you ever want to come back to us and say, just prove to us we hit the substrate, that would be one assay. Okay.
Have you done the same assay with the C2-methyl on the pyridine, so the lead compound?
No, I think this would be the only compound we've done this assay. I think the ALK5, we tried to do ALK5 with the phosphosMAD2, and that didn't work, but maybe we can try and get better sensitivity on the antibody.
Okay.
The final assay was then looking again at the transcriptional reporters. These assays have been a bit more noisy. The ALK5 assay is working really nicely in hex cells. So again, you can get quite the distinction there. In this particular experiment, something didn't work. The signal is very poor. And we've had, you use your batch of growth factor, and then we've certainly run into times when the growth factor seems to, but it's been in the freezer and suddenly it stopped working. In theory, they're all aliquoted and we don't quite, but that happens. And then sometimes, if you pathologize your cells a few times, it stops giving its robust signal. So, but I pull that in because the IC50 we got on this day was actually exactly the same as a kind of IC50 with a better response. So those assays are working, but we really like the nanobret assay. It's very quick, robust. We can do it in a 384 well plate, for example. So these are data from 384 well. This will be our concentration of tracer. So these are just independent experiments. So this would be a triplicate, a technical triplicate, and another technical triplicate as an independent. These would be the different IC50s on those 2 days. They're not too bad. I think the curves are quite nice. So at the moment, we really have this up and running for ALK2. We haven't really started it for ALK5, ALK3, ALK1. So that might be the goal for the next month. Promega are actively helping us with that. Okay, I won't dwell on the IC50s here. I'll come back to those. Okay.
The data you sent to me this morning, is that ALK5 data in the nanobret?
No, that's all ALK. Oh, that would have been luciferase for the ALK5 and nanobret for the ALK2.
Okay,
yeah.
okay.
So that would be...
Okay, good. That's good
this,
to know. Yeah.
That would be this data for ALK5.
Okay.
And this would be the nanobret for ALK2.
Okay. Perfect.
Okay.
How do you pick your tracer concentration? I see that it also varies.
So we've been discussing that with Promega. So they suggested a tracer that's in the kind of midpoint of the EC50 curve. So we would measure a curve just for the tracer alone, no compounds. Get an EC50 for the tracer, and then I can't remember whether it's around 0.6 or 0.5, in the transition. I think that's probably on their website as a guide to the assay. It's published. So what we had trouble with is that we didn't really know all the intricacies of the assay. So I should say we received the nanobret plasmids and tracers about 3 weeks ago. So subsequent to the last meeting, and of course it takes you 2 weeks to learn the assay. So we only collected the data probably, Friday, and then analyzed it on Monday. So I've only had this data in the last couple of hours to kind of put together these slides.
I didn't appreciate that.
So one of the things we were looking at was, we originally started with a 96 well plate, and when we transferred to a 384 well plate, the IC50s just went completely haywire. And it turned out we had to adjust the tracer concentration. So we've got 2 micromolar tracer, 0.65 micromolar tracer. So that makes a huge difference. And then we also had a difference between MBS plates, like non-binding surface plates, and polypropylene plates. So when we switched the IC50s, there was a sort of 2-fold difference. So we're actually getting, when we use the MBS plates, probably these IC50s are slightly better, but we didn't have any 384 well MBS plates. Okay. So
it's still not locked, like you're gonna...
Well, no, I think we'll run with this.
Okay.
Yeah.
You don't want to change these plates then? You're comfortable with it?
Yeah, I think so. But I need to go back and talk to the postdoc because, when the optimization is just testing 2 or 3 compounds, and now we're switching to 30 compounds, so we can check, have you changed your mind up to 30 compounds compared to, 2 compounds? Okay. I'm having trouble rolling them, changing. Okay, just okay. This is what I was pointing about, the 96 versus 384, so you can't quite see the bottom. So some of them are not too bad, these 206, 255, 355. With this one, typically we had around 25. And this is 34, but it sometimes went to 45, so we're kind of seeing some difference. Okay. So the aims we had were to test these 30 compounds and start getting our crystallography going. So we had the data that I've given Jeff is basically the nanobret for the ALK2 and then luciferase reporter for ALK5. They're both in HEC293 cells, and at the moment these are all wild-type assays, so we don't have the mutants, but again, that will be data we'll generate for the next meeting. Because I received the data yesterday, I still haven't maybe put this into tables and SAR that can really be helpful today, but maybe if I just run through it in like 2 minutes really quickly.
Yeah, I've got it.
And then we can catch up later as well.
Yeah.
I also want to proofread that we copied all the numbers and the data for the right compounds. So if something strikes you as really bizarre, just hold fire.
Yeah, no, nothing jumps.
I
seem to line up.
I found one compound where I do see a suspicion, but the rest I've sort of checked and I think it's okay. So this was the controls of 24 nanomolar and the selectivity over ALK5. Some of the compounds were quite weaker. I put a note here where I thought there was agreement between the urethins data. I didn't have time to check the reaction biology, but that's the next step. Jeff, you probably already did that.
I've got it.
Yeah. Next slide. Again, these ones were in agreement with a slightly weaker compound. So this is the pyrazine here. This compound was one I noted there was a shift. We can see how the reaction biology data, which I didn't actually have time to look at. So I thought there was quite, with the others, is, you sort of go from 20 nanomolar in the in vitro kinase to 89. I accept that, but this one seemed to be a bit bigger than I thought. So whether we made a mistake, who knows? But we can, if you want us to double check a compound, we can repeat the assay. Okay. This one again seemed to agree. You can see there's virtually no inhibition here, and I think it was a really poor, like a greater than 1 micromolar inhibitor, but it retained ALK5 activity. And according to Greg's comment, there's a chance that this is interfering with the alpha C in position. This one again had a slightly bigger shift than I would have expected based on the in vitro kinase. This one again had a big shift. This one was slightly bigger shift. So there's a few here where the cell activity would be something we can repeat or kind of think about.
Yeah, I think it lines up pretty well with the RBC.
Okay, yeah. And then at this point, I've run out of time to kind of look at the figures, but this one again had quite nice activity, 58 nanomolar, but then you would expect that. All we did is change this ring. Some of these I was hoping with this one, I thought it was from the in vitro, I was quite optimistic with it. Again, that's gone a bit weaker. This one actually was still quite potent with the linker. This one again, where we've changed the methoxys, we lost the cell activity, which again was disappointing. Maybe we can see how that compares with reaction biology. This one, again, we didn't get any nice curve. Again, no inhibition. Again, weaker. Not too bad. Okay. Again, this one's quite nice.
Yeah. That's definitely very nice.
But again, as Greg was saying, larger groups up here, many of those have failed. Overall, I think the ALK5 data is quite nice in picking up high IC50s. Again, this one's very similar to the other one, isn't it?
So you remind me, what would be our cutoff in solid assay for selectivity against ALK5?
I think it's more of a ratio at this point. So right now we're just gathering data. So we haven't set a hard criteria at this point. We're just kind of benchmarking everything to see exactly where we stand.
I think 30-fold would be very easy, and 50-fold should be very possible.
We set a high bar for TPP, but as Jeff said, we'll start from where we are. The graph is about 3 micromolar.
So this is our most potent compound. This is a reference compound. This is one of the ones that Greg had synthesized and published. So all this does is just return the amine group here, and you can see now we're down to 5 nanomolar. So that's the difference between a hydrogen or a metal and amine. This one is surprising. I think we published better cell activity than that. So I can double check that one. Maybe that's one way I've made a cut and paste.
That was the one where,
Okay.
I think the ALK5
Yeah.
potency was more potent than the ALK2 for that.
what I wanted to do is start making a table where we use different assays. So, for example, the phosphorus-MAD immunofluorescence, the nanobret, and luciferase, and kind of collect where discrepancies lie between the assays. This is a work in progress, and this is when we were still doing our optimization.
Yeah.
So maybe next meeting we'll also fill this in a little bit for you too. So I think I'll stop there. These are the guys. John Food is most of the work. Okay. Okay. Your mouth. Any questions while we change slides?
It's not about your presentation, it's about the RBC data or cross-section data plotted in Oracle, and I saw just for some compounds, not for all compounds, activity goes almost up to from 300 to 0, not from 100. There is some either noise or how they normalize data for plates, and they either recommend to retest those compounds or those compounds have to be repeated individually.
I think, I saw that, I looked at it, I think predominantly it was in one of the mutants, the 328B mutant, where we saw that issue, where, the potency isn't as good in that, against that particular mutant. So I'm not sure, RBC agreed to test that mutant for us, because it is important in DIPG, but they did say that the assay is still under development, and it's not available to everyone. They did it for us, so, they reported the data, and I'll show it on my slides as well, the IC50.
I don't know if reaction biology are on the call. I know we still need to connect with them to try and see that we can help them from our side in terms of providing new constructs for their assays.
Yeah, I didn't see Kate on the call.
Okay, we can also try, if they're struggling with one of their proteins, we can also send them protein, or just to see if the assay is robust with different sources.
I'll just follow up with RBC.
Yeah, okay.
As an action item. Okay.
One quick question actually. I don't know if you're going to speak it. The NH2 compounds that are so much more potent, do they have good CNS penetration? Have we looked at that yet? Or do we actually take a hit? CNS. Cell permeability.
We would have, ITR would have done some assays on that. I've shared that with Jeff.
Okay.
Off the top of my head, I don't know. I know. Yeah, I can't answer that.
Okay. Just trying to pull up the next presentation here. Just give me 2 seconds. Okay, so I'm just going to give a very quick presentation. I think Alex went through a lot of this already. So, but I did want to bring up some of the old data and talk about some of the in vivo data that's been generated. So again, we're having trouble. So, as was mentioned, we tested a group of 30 compounds against these mutants, the DIPG as well as the FOP mutants, and against ALK5. And then moving forward, because the data against the mutants was consistent with what we saw against the wild type, we're going to test compounds against ALK1, ALK2, and ALK5, the wild type, and then spot check against some of the other kinases as we move forward.
Sorry, I forgot what the position for ALK1 selectivity-wise. What are we trying to get?
Well, ALK1 we don't know really yet. We don't have a ton of data. This is something that we had a call with Boehringer Ingelheim. They mentioned that ALK1 was a real issue for them in their ALK2 project. And we've seen
chemistry or an issue actually in mice or something?
No, it was more of an issue just to get selectivity, right? So I don't think it's a big issue if we hit ALK1, but I think, if we can try to keep the phenotype, ALK2 related as much as possible, I think that would be a good goal. So, Alex showed some of the not the RBC data. Basically, the SAR trends were maintained with what we saw with the Eurofins results. There was some differences in absolute activity, and there were a couple of compounds that didn't line up perfectly. And the one difference was that RBC ran the compounds at an ATP concentration of 10 micromolar across all of the assays, whereas at Eurofins they were using the KM, which is slightly different between the different kinases. That might explain some of the differences we saw, but I'm not that concerned about it, and certainly the assays that RBC developed against the mutants were optimized at 10 micromolar ATP concentration. So, I think we'll continue moving forward with that.
Is there anything known about the effects on the KM with the mutants? I mean,
yeah, we had in one of the collaborators, Greg Paul Yu who was at MGH and now at Brigham. He worked out a radio-labeled assay for ALK2 with a student, and they'd measured some KMs. We looked at the mutants and the wild type. I think we published that. There is small differences, maybe twofold, threefold, but they're all within the kind of 10 to 30 micromolar range, as far as I remember.
Okay, so I have assembled some of this data into this spreadsheet. I'll walk through it quickly, and because we're getting a little bit shorter on time, I'll just highlight some of the key compounds. And I guess I just learned that this is actually the luciferase, the ALK5 data, so I'll update this as we move forward. So the aminopyridine compound, M4K 1046, is very, very potent, but not very selective against ALK5, and this translates into what we see in the cell-based assay. There's only an 8-fold difference between ALK5 and ALK2 in this cell-based assay. And this lack of selectivity was the primary reason that the lead compound was this methyl compound, M4K 1055. However, in this set of assays, this compound really wasn't that potent. It's around 150 nanomolar at RBC against the ALK2 wild type. It's selective, and again, that translates into exactly kind of what we see in the cell-based assays. Same kind of ratios, and the compound's stable, shows very good PK, but it's not that potent against ALK2, or not as potent as we'd like probably. And that may be one of the reasons that, and I'll highlight some of the data, that we didn't see much of an effect in vivo with this compound. Even though the compound had great plasma exposure and great brain exposure, we didn't see a PD effect, and we didn't see much efficacy. So anyway, we'll talk about that. The chloro compound was not well tolerated, so I won't go over that. I think it's pretty clear, the chloro here. When this is a hydrogen, the compound is actually well tolerated. It had quite good PK as well, and it's reasonably selective. Certainly less selective than with the methyl, but we do regain 5-fold in potency, and we still have a 40-fold window with respect to ALK5, and that continues into the cell-based data, so that we still have a 40-fold window, and it's a 20 nanomolar compound. So this might be a really good compound that we can put in vivo, and we will actually. We're scaling it up to get some plasma exposures in our hands for this compound, as well as for this compound, so that we can benchmark our in-house data with what's been reported. And but this might be a really interesting compound that might show better efficacy than the M4K 1055.
The way you phrase that, should this be our lead, what are you looking for before you felt you feel confident on that?
Well, I'm just saying, we're going to continue doing SAR around this scaffold. We had originally planned to do most of the SAR with the methyl in place to maintain that specificity, and to spot check with the hydrogen from time to time. What we might do now is focus more on the unsubstituted, and then spot check with the amino and the methyls.
Right. That's effectively the strategy that Paul had taken in Oxford over the few months leading to this grant. Yeah. Right.
Yeah, and so I think that's most of the SAR that we have in the rest of the table has only the hydrogen. And we've made some compounds that we've submitted for testing that have the methyl in place with some additional SAR, and we'll see very shortly whether or not that loss in potency is consistent. And since it is a hinge binder, I suspect it's probably going to be pretty consistent. But we'll get the data, I think, in the next month to be able to really firm that up. But so, I think generating this consistent set of data is really starting to pay off already. Let me just highlight a couple of other compounds because Alex already went through this. We think this set of compounds here is fairly interesting, where you kind of extend the water-soluble group out a little bit. It's not that potent in the cell-based assay. This compound, that might just be solubility. But when you have a more solubilizing group like this analog, 1134, cell-based potency is quite good, and the selectivity is around 20-fold as well. So reasonable starting positions for SAR. Some of these compounds I think you talked about. But sorry?
Keiko is missing.
Yeah, we only have Keiko for the first set of 14 compounds, and we're really focusing on the MDCK and DR1 for this project. So I think that assay is coming up, and we'll incorporate it here. But but I mean, if you think we any flat, some of the compounds, the ones you might expect. So these ones that have the really good PK have really quite good Keiko and no efflux. The amino compounds have reasonable Keiko but strong efflux. So that's always a balance that we'll have to continue to monitor. But it's I think we'll get Keiko for the most promising compounds, but probably not all of them. We'll let MDCK and DR1 drive it. There's a couple of other interesting compounds that we just wanted to highlight. 1212 and 1216. So moving the piperazine from the para position to the ortho position really gives quite nice compounds with 80-fold selectivity in the enzymatic assays and 30-fold selectivity in the cell-based assays. So this is a really interesting compound that we're going to remake.
Do you think that would be compatible with the sulfonamide or these ether-phenyl things that Greg was talking about? Or do you think
on this side of the molecule?
Yeah.
Yeah, it really could be, and that's something
there could be a clash there. That would be good to model.
Yeah, and this is one that we want to get an X-ray of. This is one that we'll flag. We want to remake it to get some PK. I think, depending on the pharmacokinetics of this compound, it could be a really good tool compound as well to look at in some in vivo experiments. You could imagine combining both the ortho and the para substitution to try to pick up some potency or selectivity. And then, the trimethoxy SAR. This compound is stable, but when you N-methylate it, it's not, and that could be the effect of the trimethoxy coming out, where in some cases this is tolerated and it's stable, in others it doesn't seem to be very well tolerated.
Could be in the method,
it could be, yeah.
We use all the CNS. Sometimes actually for CNS penetration, the N-method actually affords that rapid absorption, enough to get penetration into the brain. So that actually may not be bad in terms of CNS uptake.
Yeah.
So that's something to monitor. With methylation, that could kind of be a project into the brain.
Yeah, and we could also look at other substitutions there too, right? Yeah. So we think this is probably among the most interesting results that we were able to generate.
It's a little better in the M&M, right?
Yeah.
Than the other one.
Yeah. So this one we're interested, but because of this, we won't be able to get in vivo data. I mean, we could, but I think let's use this data to prioritize.
So those
compounds.
This one is the one you're going to.
Yeah, and the lead compound as well, just to benchmark in our own hands. Yeah.
Actually, also 1215, it would be nice to follow the metabolite. Is that?
Yeah, we could look at that. I think we generated this data in-house, so we could go back to our MLM data and see if it is N-demethylation.
And you have the compound.
And it's right here. Right?
Both are there,
Yeah. So, and then this is just the lead compound, or the compound that RBC uses as a reference in their assays. So we got the data for that compound as well. And actually, this is the compound that I'll show you in the next part of my presentation, where I talk about some.
That's the compound Greg presented in his first slide.
Exactly, right? The kind of lead. And this one actually showed. Sorry, yeah, 1016, yeah.
1016, yeah.
Yeah.
It's 193189.
Yeah. Yeah, Jeff.
Survival.
Okay, so I just wanted to go through this quickly as well because I think it's important to have everyone on the same page about the data that we're generating and trying to understand the in vivo data that was generated. So this is the literature data for these compounds with NH2, hydrogen, and methyl. You can see that originally it was reported as having a 24 nanomolar IC50 against ALK2. We get a little bit higher. It's N equals 1. We're going to retest it. But, you know, the cell-based activity stays, you know, in this 100 to 200.
That's not unique to this compound. Every single compound is much higher, but that was a radiolabel assay. Probably the ATP concentration might have been different.
Right. And so, you know, I mean, this is why we need to get multiple N values for some of these.
But the reactionology is radiolabel as well, isn't it?
Yeah. And then these compounds were tested at ICR for their mouse PK. I've labeled them here. I haven't put the M4K numbers, but basically because they're not in the table. But what you can see is that the hydrogen analog 844 gives really good exposure. When dosed 5 mg per kg, you get a Cmax of around 1.2 micromolar. Clearance is reasonable. Half-life is okay, and the volume of distribution is on the lower side. The amino compound here shows, again, reasonable PK actually for a compound that shows pretty high efflux, but you know, it's certainly a little bit lower than the methyl compound, than the hydrogen compound. And then the methyl compound, which is the lead, shows, again, around this 1 micromolar exposure Cmax with a better T-half than the hydrogen compound. But still, both of these compounds are quite good, and the hydrogen is quite good. So this goes into, you know, which SAR series should we follow, given that, you know, the hydrogen analogs are more potent and show a PK profile that's similar, if not superior, to the methyl compound. And when these compounds were moved in vivo, so this is the methyl compound, into these efficacy studies in this DIPG 007 line, which contains the FOP 206 mutant, the LDN compound gave 26-day increase in survival, whereas the 117 compound only showed a 15-day improvement in survival, which is, you know, marginal, I think. And importantly, when I saw this data, I didn't appreciate it at first, but as I've kind of gone back and looked at everything in light of all of the data that we're starting to generate, so this 189 compound, the compound that showed the 25-day improvement in survival, showed very little weight loss over the duration of the experiment, a little bit at first, but it was reasonably well tolerated. Showed pretty good plasma concentrations of around 2.5, 3 micromolar Cmax, which is shown here. And also, it penetrated the brain. 2 hours after a 35 mg per kg dose, there was about a 3.5 micromolar concentration. And that level with that compound impacted the phospho-SMAD and the downstream effect of the expression of ID1, which is what you would expect from an ALK2 inhibitor. Whereas the 117, which is the methyl compound, was really well tolerated, showed exposure similar to what we expected to see based on the pharmacokinetics that we had already got. And in the brain, we had 12 micromolar of this compound 2 hours after a 25 mg per kg dose on day 14. But we had little, if any, effect across the 3 mice on the phospho-SMAD, and the same observation for the downstream effect on ID1. So, you know, that the relatively low efficacy that we saw with that compound likely is explained by, you know, the fact that it's probably just not that potent. and not as potent as we would like it to be, or as much as we need it to be against ALK2.
So in the in vitro phosphoramide, it looks good?
No, not against this line, which is the 007. You can see even this is the in vitro. So you can see this compound affected ID1 all the way across in all of these cell lines, actually. Whereas this compound, this is the cell line that the efficacy was ran in, we're not affecting ID1. There might be a little bit of phosphosmad at the highest dose, but we're not really affecting the ID1 there. Whereas
that would be a criteria before you advance other compounds.
I think so. Yeah, that's basically wanted to show this. We need to show that. Yeah, we need to show that functional pharmacodynamic effect before we move a compound in vivo into in vivo efficacy studies. And in fact, we do see the effect of ID1 on this cell line, right? So it might be effective against an efficacy model based on this particular cell line. And you can see actually that this was borne out in the cell viability experiment as well, so the cell potency. This compound was active against all of these, both mutants and the wild type cell lines that contain, so this one has this 206H mutant, and the 117, you can see that for this mutant for which the efficacy study was done, the potency in is really not that great. It's out around 5 or 6 micromolar.
The other way around, isn't it? I think the curve on the left is the R206H, and the other one is the G328.
No, I don't think.
No.
No. You see, this is the squares.
Oh.
I think so.
Okay.
That's how I read it anyway. But
oh, interesting.
yeah. See, these are the circles. Anyway.
Hard to read, actually.
Yeah, it's hard.
Yeah.
But basically what I'm thinking is, this data all makes sense now based on what we're seeing.
Can you tell better by the colors rather than the shape? Maybe not. Okay.
Nah.
All right.
Yeah. Anyway. So what else can I show? So basically, this weaker potency against this particular cell line might explain what the relatively poor efficacy that we saw in that experiment, and getting a compound with better potency to move into these models. I think we should start to see something as good, if not hopefully much better than what we saw with 189. And with that, we can move to
So that arrow, yeah.
I think it's trying to save. There we go. Move to David, and he can tell us a little bit about the chemistry that's been ongoing here at OICR.
Perfect, thanks, Jeff. So this was just a recap from last time and from the CTIP. So we were seeing our chemistry efforts were focused on putting a methyl group in this place and just doing some different substitutions to see how well that's tolerated, trying to increase the potency of our pocket side group and replace the trimethoxyaryl, and then looking at some different substitutions on the solvent side solubilizing group to try and get better potency and selectivity against ALK5. So the initial data on this compound, I have the RBC data listed here. It was similar to the Eurofins data, although RBC did show greater selectivity. The ALK5 was much weaker in the RBC assay, and we're kind of seeing that in the cell-based assay that Alex did as well. It looks pretty good, and the RBC seems to correlate better with the cell-based activity. So looking at a couple of the key compounds that Jeff was talking about, moving the piperazine group to the ortho position is well tolerated and gives good selectivity, actually 80-fold selectivity in our biochemical assays. That seems to erode a little bit in the cellular assays to a little bit worse than 1062 there, but not so much. They're almost equivalent in terms of selectivity in the cell-based nanobread assay. And then this compound looked really good be doing some substitutions out here. All the mutants seem to correlate well with the wild type. These compounds were some where we saw good data from urofins, but in the RBC assay and now in the cell-based, were very poor. So in the cell-based data, these were totally inactive, where we were seeing good cell activity at one point with the urofins data. So we might follow up on a couple of these compounds just to confirm that result, but it seems like the methyl is really ruining the cell, or ruining the potency at this position. We can always spot check with our best compounds, so if we get a good jump in potency. So based on what we were seeing from the urofins data, our chemistry plans were moving the methyl around, seeing if we could change some torsional angles. So here we have M4K 2009 and 2025, just seeing if changing the angle on the methyl will actually give us a jump in potency. You can see in the picture, it's really, both of them are rotated quite a bit. We're also exploring different pocket side groups. So it was interesting to hear Greg talk about the importance of that methoxy. We have quite a few analogs with that, but we've also removed it in a few. So we'll see if what the trend that he was seeing continues. And we have some bulky groups jutting out, just like he was seeing with the benzyloxy sulfonamide. So that'll be interesting to get that data back from RBC. We've also been playing around with some of these solvent side solubilizing groups, trying to get increased potency. So David Smil had made the meta piperazine as well, so we'll see if any of those ortho, para, and meta are tolerated, give similar data, or if there's any kind of effect there. And then changing the size of the ring, so here in indazole. We also have been scaling up a few of these compounds for in vivo PK studies. So we've been making these 5, and those will be ready as soon as the mice are in a couple weeks. Ready to go. And based on that Novartis patent, compound 34, I know Charles River is working on making a couple compounds from this patent, but we also want to explore just different orientations of this amide in a couple select examples, just to see how well that's tolerated, and maybe with the methyl there. Since we were getting good kinome-wide cell activity with that methyl group in place. So if we can have it and restore some of the potency, that would be a real benefit.
Sorry, can you go back one slide? You said you're scaling up. When you say scaling up, to how much?
200 milligrams?
200 milligrams.
We have plenty of the starting materials, so we just want to make sure we have enough of these to profile through whatever they may need to be.
We're doing that in terms.
Yeah.
Scale up.
Just 200 million.
One in the middle was already done at ICR. So are you trying to duplicate some of the ICR data for the PK?
Yeah, well, we run a slightly different version of the oral PK in-house. So we need to benchmark what the results that we get internally against what was generated before, so that we can compare as we do the SAR.
Because ours is more of a rapid PK,
Yeah.
not a full PK.
Right. We just look at exposure at certain time points in the plasma, as opposed to getting the full pharmacokinetic profile. We'll outsource that for the best compounds.
That's something to realize also the PK, even the IVR at 5 mg per kg, that might have to be, depending on different compounds, 5 mg per kg might be a bit
too high for the first.
1 or 2 mg per kg might have to.
All the ICRs.
Yeah. Usually we don't have exact same for IV and PO. That's what was done before. Normally you do higher PO and lower PO, right? So.
That also high, sometimes you're going to mean for the exposure that you're getting right into the plasma for those types of doses is huge. So
that might be why some of the N-methyl analogs were toxic,
Yes.
right? In the IVR, is when they might have just had great exposure and
Exactly.
it went through the roof at 5 mg
Plus,
per
too,
kg.
if you got compounds that are not very solid and stuff, you go into the splash out and get all kind of effect.
These are some of the compounds we've made. Some of them have been sent. Some of them are waiting to go to RBC in the next batch. And the ones that were sent to RBC have also been sent to FGC Oxford, Alex, for cellular testing. These are some of our future chemistry plans. So we can't really discriminate at this point between the parapiperazine and the orthopiperazine. So we're going to do some substitutions using both on the intermediate phenyl ring, as well as some substitutions on the capped piperazine with different electronic groups, as well as sterically bulky, so adding some methyls, maybe we change the PK, and then capping it with some different groups that might help the stability of those compounds, change the MLM, HLM numbers, and get a better compound. And then just some more general ideas. I mean, we're working on changing the pocket side binder slightly, introducing amides into there, just looking, spot checking those compounds, just a couple, putting groups off. And then we're also interested in cyclizing around there. It seems like the bulk is well tolerated from this piperazine, and we might get some good selectivity that way too between ALK2 and ALK5. By introducing another cycle in there. Those typically show good CNS exposure as well. So just ripping off of this compound in particular. So we've synthesized 35 plus compounds to date. Most of those were answering the questions that I outlined at the start regarding the methyl, moving the methyl around, pocket side and solvent side binders. 25 were sent to RBC in February, and we hope to send around the same number for March. Those same compounds were submitted to SGC Oxford for cellular-based assays, and are going to be studied in ADME here at OSDR. We've started doing some modifications around M4K 1212, that's the orthopiperazine. We're looking at the amide, similar to Novartis compound 34, and we're nearly done the scale-up for in vivo PK studies, rapid PK.
So I wanted to the compound progression discussion at the beginning, but it's going to be routine to send all the compounds to the cell-based assays?
Yeah. Yes.
That's great.
Yeah. When we send them to RBC, we'll prepare the samples and send them to Alex at the same time. Yeah. Okay. Sue? Are you on the line still?
Hi, yes, I am online. I just the computer went blank just for a moment, sorry.
So I've got your slides on the screen
great.
there now. Hopefully you can see them. I can advance them for you. Just let me know.
Please. Yeah. Okay. So if you go first to the next slide, please. Okay, so I've just given a general update here of where we're at. So we've continued doing some chemistry during February, but as we've discussed, we have a very high current workload here, and so synthetic progress has been a little slower than we had originally planned. But we've completed the synthesis of the Merck patented compound, and that's actually currently being purified, so hopefully we should have that fairly soon. The two sort of right and left-hand side parts of the blueprint patent compounds, we've got the core. On a small scale, we've got to make a bit more to be able to make all the targets that we required. We've made the aryl bromides. Sorry, I haven't got any structures here, but I made the aryl bromides required for the coupling. As far as the targets within the pyridine series, as you know, we've got two ready for screening, but we've made limited further progress with those targets. There's a little bit here and there. In the meantime, we've actually run a couple of brainstorming sessions at our two different sites at Charles River. We did present some preliminary outcomes from those at the last TC last month. But we've done a bit more analysis, and I think some of the results that have been presented today may change some of our thoughts. But we have noticed that some of our ideas do overlap with the ones that David has just presented. So, you know, we will obviously, as Jeff said at the beginning, discuss. We won't make any compounds until we've discussed who's actually making them. So we've got a whole raft of other ideas that we're still going through the docking and looking at how well they might fit into the pocket. So we've also done a little bit of modelling in terms of an alignment of ALK2 and ALK5 to try and understand if there was any real good reason for the selectivity of the methylpyridines. But looking at some of the data, it looks like we still get quite good selectivity with the H-pyridines. So, yeah, we need to have a little bit of a think about that as well. So the other thing that we've done, as Jeff mentioned at the beginning, we've now got an e-room set up using the CRL portal. So at the moment, Jeff has access to that, as does Owen and Arij. So we can extend that to other collaborators as required. But it's quite a nice way of sharing documents and making sure that we've all got the most up-to-date version of all the information. So I'll go on to a little bit more detail in some of the areas. So we can go to the next slide. Yeah. So we discussed this patent that was published in January in the WebEx last month. And as we highlighted at that time, compound 34, shown in the middle there, was considered to be the most interesting compound because they had actually done a lot of further work with that compound. They described a lot of additional work. But actually, when you look at it, it's got, 4 H-bond donors, it's got a high C-log P, it's high molecular weight. The likelihood of that being brain penetrant is very low. So I went through the patent in a little bit more detail since that last meeting, and have identified a couple of compounds that do look interesting that we're going to try and prepare. The compound 45, so on the right-hand panel, if you look at the compounds where R is at NH2, these two compounds, as I say, R is NH2, look to be quite potent at ALK2 as described in the patent. And they've got moderate MPO scores, which could mean that they should be more brain penetrant than the compound 34. But also think that it will be worthwhile making perhaps the methyl and maybe the H from what we've discussed earlier today, maybe the H compounds as well, because they will definitely have a higher MPO score. I think you mentioned about the high efflux that's observed with some of the aminopyridines, so these compounds should have a better ADME profile as well.
Right, yeah, no, I think that's a good idea, Susan.
Should still evaluate. Is that like an internally hydrogen bond? I don't know if that's picked up in the MPO score. We should have
I'm sorry. So could you repeat that? I didn't quite catch it.
Yeah, I don't know how well the MPO score captures the ability of these compounds to internally hydrogen bond, but we should at least make one example to compare.
Definitely. Oh, we will definitely make the compounds with the NH2, because obviously, as you say, you've got the potential for the internal hydrogen bond, so it may only be one of those donors participating. But even so, you've still got 3 donors, which is quite high for a brain penetrant compound. But I agree with you, there potentially is an internal hydrogen bond there. Okay, so if we move on to the next slide. So as I mentioned, we held 2 brainstorming sessions at the end of January, beginning of February. We discussed the preliminary results last month. We've docked a number of selected ideas, and obviously we will continue to look at further ideas, but we've prioritised those that look look to us to be the most interesting, and that's what I've actually got in this presentation. There are quite a few others that we can come back to when we've done a bit more docking or when we've got a few more results from some of the other compounds that have been made. So if you move on to the next few slides, actually summarise some of those ideas. So a lot of the ideas that we had when we held the brainstorming session were around how can we replace that trimethoxyphenol, which everyone is a little bit nervous about, whether it be due to microsomal stability or potential for CYP liability. Ideally, we might want to remove at least one of the methoxy groups. So some of the ideas on the top panel looking at replacing one of the methoxy groups. However, from what was said earlier today, it may be the wrong methoxy group that we're actually trying to replace there, because it looked like one of the metamethoxys was more had a greater potential to be replaced. But I don't know how many of these sorts of things have actually been looked at to any great extent. So some of them are looking at extending a bit further. So a lot of the compounds, it looks like there's a water-mediated interaction between the methoxy, one of the metamethoxys, and the lysine. Excuse me. And things like an amide or the THP on the right-hand side may well extend directly to pick up that lysine interaction. And there's a similar thing with a nitrile, which is in the second panel down. But I think these are quite similar to some of the things that David has spoken about. So we'll probably hold on any of those until we've got a bit, until we've got a bit more data on some of those compounds. Mm-hmm. So if you move on to the next slide, please. So we've seen a number of we've seen a number of compounds where the two of the methoxy groups have been replaced by pyrazole ring effectively to give the methoxy indazole. And that seemed to retain activity, but I'm not sure that it actually retains the cellular activity. But it may be possible to change that slightly and have the midazole instead of the indazole and pick up the interaction with the sort of nitrogen, the sort of para-nitrogen. I'm going to have to have a small water, sorry. And there's some other benzimidazole type strong compounds or a pyridyl. Excuse me. There's also things like the quinoline compounds in the panel below, but I think some of those may have already been made. But I haven't seen results for compounds, actually a direct comparison. I believe they may not be these direct comparisons, but different modifications at the other end of the molecule. So if you move on to the next slide. So we've looked at actually replacing the phenol ring by some heterocycles, and one of the things that looked quite interesting was to replace it by pyrazole ring. And you can still reach into the pocket to get some reasonable interactions. For example, compound B in the panel at the top, that hydroxyethyl appears to interact with glue 248, as does if you look at the amide at compound C in the panel at the top. It's on the modelling on the right-hand side at the bottom, where you can start to see some water-mediated interaction with both the lysine and that glutamate. So there's some interesting compounds there that might be worth considering. We also looked at some amides. With the methyl group in place, we found that the amide with the carbonyl next to the pyridine ring, actually you got a clash. But if you do it in the desmethyl, it's potentially better. But if you turn the amide round so it's N-linked onto the pyridine, you can get a reasonable interaction. And so then the carbonyl is in a relatively lipophilic region. So there's some possible alternative rings there. So moving on to the central part, I'll try and speed up as I can see the time's disappearing rather rapidly. So on this slide, we were looking to modify the hinge binder region. We were sort of quite intrigued by the effect of this methyl and whether you could combine it with the amino group to get the increased potency associated with the amino group, but actually also get the increased selectivity that we see with the methyl. We were also looking at, I know you've made the pyrazine, but I don't believe you actually made a pyridazine, and one of our chemists had experience using that in one of the series that they'd worked on as an alternative replacement for the pyridine. And then there's a couple of things that sort of link into the quinazoline series that the guys at the ICR are working on. So rather than having quinazoline, have pyrimidinone or pyridone at the hinge binder, and also the possibility of having an aza-indole, which is quite a common hinge binder, which again picks up bidentate interaction at the hinge, and still gives you a similar approach out into the pocket. Okay, so move on to the next slide. Some of the ideas that we had around replacing the solvent-facing end were to replace the phenol ring by a pyrazole. I think that's been done in some of the literature compounds, but I think there's only relatively limited exploration within our series. It does provide a reduced lipophilicity, but it still maintains a good MPO score. And then, again, in the same way as I think David has just spoken about putting linkers or substituents into the phenol ring or onto the pyrazine. So at the moment, we are fully occupied with trying to prepare the patented compounds or the initial set of compounds. So if there are any of these ideas that you guys at ICR are interested in picking up, please do feel free to let us know. We also had, as part of the brainstorming, was how might we identify additional chemical matter if we do need yet another series. So there's options of potentially screening the soft focus libraries, but that would probably require additional biology resource. But we could use virtual screening to prioritize. There's a wide range of known lysine binding groups, and we could look to explore that through doing some library synthesis. I don't believe macrocycles have been looked at in the quinazoline. I know we're trying to prepare some of the macrocycles in the pyridine series, but I wondered whether there were some opportunities there. We've looked at what is actually driving the selectivity for ALK2 over ALK5, and we've been trying to do a little bit of sequence alignment, which is on the next slide. And it might be possible to actually introduce selectivity from that. So on this next slide, we've got some preliminary information. So there's a very high homology between ALK2 and ALK5. It's about 82% in the binding pocket. The major differences really are the gatekeeper, the threonine in ALK2 and serine in ALK5. We were wondering whether that methyl group on the threonine was actually what was giving rise to some of the selectivity, because you might think you might need to prefer a more lipophilic group rather than the amino group, which is obviously going to interact with the OH. And we're trying to do a little bit of docking, but so far we haven't actually been able to show anything, so I haven't got any pictures to show yet. But there are a few differences in the solvent exposed end of the binding pocket. But I was intrigued by some of the literature. I think it was one of Alex's papers that suggested that the hydrogen bonding network between the isoforms changes, and that might be quite difficult to predict. I think that's my last slide. Sorry, I've whizzed through that a little bit quickly because I realised that we're running very short of time. So apologies for that. Are there any questions?
Thanks, Sue. I think that's great. I think, there's some really interesting ideas in there. I think, over the next couple of weeks, we'll try to set something up so we can go over them as a chemistry team, perhaps, and try to
I think it'd be good to have perhaps have a chemistry WebEx in a couple of weeks' time.
Yeah, absolutely. I think I'm away for a week after this meeting, but definitely before the next team WebEx, we'll set something up with just the chemists so that we can finalize some of these plans and prioritize amongst them. And hopefully we'll have some of the RBC results on the 25 compounds that we've submitted to help with that discussion by the time we have it. Okay, but that's great. So it looks like we don't have a ton of time for the administration.
Yeah, no, I'm just going to take, thanks, Joe. I'm just going to take two minutes very quickly to go through a couple things. And first off, the team from Charles River, thanks very much. I think what we will do is we'll take a look at the timing that we set up for these meetings, because we rushed through quite a bit of information here from the Charles River side, which I think we could have spent more time really developing in and thinking about. So we have to make sure that we allocate more time. But thanks very much for this presentation, and we'll work through that. On the other administration items, very quickly, the two that I just want to touch base with, Charles River, as mentioned, they've set up a data room or data site. We're going to roll that out to people and get access between now and the next meeting, and deciding who has to get in, et cetera, and try and make it very available to a lot of people. The last part
if you can just give me names and email addresses, then we can extend it to additional people.
Yes. Thanks very much, and we will do that. And the next part, just as a heads up for people, we were going to talk a little bit more about the upcoming website. Arija's been working on that. We'll talk about more next time. We're running out of time here, but hopefully we'll be able to make it a central site for people to gather more information about what we're doing here and know more about, if they want to join or see some of these meetings, and maybe the coordination of this meeting went a lot better than last time, and we'll get it even better next time. So, Arija, I was going to let you talk, but my apologies for cutting you off on this. We should get back onto the time. So thanks very much.
Okay, it looks like, I think we've had another productive meeting. I think it started better this time. I think, we avoided the technical issues for the most part, other than Alexi not being able to dial in, so we'll follow up with that.
Oh, yeah, and Greg and Alexi, thanks but Greg, thanks very much for your presentation as well. It was really appreciated.
Okay, you're welcome.
Yeah, thanks, Greg, and I hope you enjoyed seeing some of the other SAR as well that we're starting to generate. So it'll be interesting to see how all of our compounds behave in each other's assays. So
I was very interested. I really was interested in that orthopiperazine orthopiperazine compound, the 1212. I'd be curious to see how that was in RIPK2, for example.
We'd be curious about that too. So that's
because we haven't really looked at any orthosubstituents, so I was a little surprised that it retained such a good ALK2 activity. So it's pretty interesting.
Yeah, absolutely. We like that one too. So, okay. So action items. I don't have a lot here. I've got follow up with RBC, with Alex, to see what we can do for them next. We want to set up a chemistry meeting before the next team meeting to try to prioritize some of these ideas. And I think just continue with the chemistry and with the plan in terms of getting a good data set that we can base future SAR decisions on, and we'll continue sending compounds to RBC. I think, Sue, we need to talk as well about getting the compounds that you do have already tested as soon as possible, so we can follow up offline there. And does anyone else have anything?
There's a Cancer Research UK Brain Tumour Conference in London in the first week of May, so our group will go to that. There'll be quite a few DIPG talks on that. For example, some experts from Toronto. I think either Michael Taylor. Yeah.
Okay.
And some other, there's a few, probably 3 people talk about DIPG. So I'll let you know what the stories emerge from there. Thanks.
Great.
All right.
Okay, so thanks everyone, and again, Sue, sorry about rushing you through your presentation. We'll move you up in the agenda for the next meeting. And thanks all, and we'll talk soon. Great work.
Thank you.
Okay, thanks.
Thanks a lot.